Hello students, welcome to EPG Pathshala again today. I am Dr. M.N. Gupta, Emeritus Professor from Indian Institute of Technology, Delhi. Today we are going to talk on the module with the title Immunotechnology. This is the first module in which we will discuss these immunotechnological techniques, and that's why we are calling this module Immunotechnology-I. There are few other modules where more and more other techniques which constitute immunotechnology will be discussed. The paper, of course, is immunology. So, starting from today, we will look at what can be called a toolbox of immunology or immunochemical techniques, because most of these techniques utilize the molecules or their fragments which belong or which are derived directly or indirectly from immune system. Then these molecules are utilized in these techniques in generating some chemistry based upon their chemical properties combined with these biological properties. So that's why these techniques are simply called immunochemical techniques. In today's lecture, the first lecture, we will focus on techniques which are essentially based upon precipitation of the antigen with the antibody preparations. The objectives of today's module is to try to understand, number one, what a variety of experimental techniques which we can use on the bench to understand the overall functioning of the immune system. By now we begin to get this idea that the immune system consists of variety of cells, molecules, and they function at various places in the body. Some are localized, some are roaming around in the body. But in experimental immunology, we have to see that what techniques are based upon the cells, what techniques are based upon these molecules, which of course are related to the immune system. So, interesting thing is that, as we will see in these set of modules which we call the immunological techniques, some techniques help us in understanding the functioning of the immune system. There is another very interesting aspect, that because of this uniqueness and a fairly specific nature of the interaction which all these cells and molecules of the immune system participate in, this can be used as a basis for generating a set of techniques which only can operate by the use of these molecules or cells of the immune system. Let me clarify what do I mean. Suppose I say that I am using a spectrophotometer to study antigen-antibody interaction. Now, spectroscopic techniques are general. They are used in various branches of the science, chemistry, biochemistry, and so on and so forth. So, that is one set of the technique, that these are, I study the interaction between the molecules belonging to the immune system or the cells bispectroscopy, so that's one set of technique. But then based upon the interaction between the molecules and cells of the immune system, I'm also able to generate some techniques which cannot be used by another set of the the molecules of the cells. Not directly, some variations is possible. So we'll try to understand that there are these two aspects of the immunochemical techniques. So how some of these techniques we will see are now the basis of invaluable diagnostic tests, design of biosensors for analytes, and to be able to detect, in many cases in a fairly quantitative fashion even, the presence of infectious agents. So the concepts which we will cover in today's module is then, of course, we will start with immunization, which basically leads to the setting where the molecules and cell of the immune system and the antigen with which we are immunizing come together. In most of these discussions in this module and many subsequent modules, all the time when we say an antibody, of course we are talking of polyclonal antibody preparations. So we will talk about the nature of this polyclonal antibody preparations. We look at how in many cases where necessary we can isolate antibodies, and then armed with that knowledge, then we will go over and look at the experimental tools related to this antigen and antibody interactions. So in many cases, the purity of the antibody preparation is simply the anti-serum which we kind of get out of the animal. In other cases, where necessary, we can actually purify this antibody to a reasonable level. Most of the immunology, of course, started with simply the use of an anti-serum, and people called it antibody preparations. Immunology started with efforts basically to understand how our bodies respond to an infectious agent, to infectious agents. And then of course we were naturally interested that how can we avoid, and if we cannot avoid, how we can minimize the consequences of this invasion. We try to avoid the invasion. Those are all preventive measures. However, if the infection does take place, in spite of the precautions which we take, then the immune system is geared towards dealing with this invasion. So we will look at the molecular and cellular consequences of this invasion. Right in the early years, immunologists developed protocols to raise antibodies in experimental animals, but they were able to do it. In the very early days, their antigen preparations were ill-defined, like some microbial preparations, killed or parts of it like a viral coat or something, of the so-called microbial antigens. But as the Landsteiner era came in, and the massive amount of the work which we did, in many cases these antigens are very well defined. We know their structure, we know what we are doing, what do we sort of can expect would be the nature of the antibody which will be created, and so many other things. So we have gained some knowledge about it, which is very helpful. So immunologists can today raise antibodies in experimental animals against known antigen preparations. That's the bottom line. The first important consideration, of course, is the choice of the experimental animal. To start with, any animal which is chosen for this purpose should be healthy and as far removed as possible on the evolutionary tree from the source of the antigen. As we learned in the module on antigens and epitopes, it is the foreignness which is an essential criterion for antigenicity. So, if the animal or the animal and the animal from where this antigen is sourced, if they are evolutionary close, then the good immune response by the experimental animal is not obtained. Also, depending upon the amount of anti-serum required, the animal with appropriate size has to be chosen. A larger animal like a horse can be bled a little bit more as compared to a smaller animal like a mouse or a rabbit. Adjuvant, the concept of the adjuvant is an important one. So let us understand it clearly. Adjuvants are substances. My choice of the word substance is intentional because I'm not defining whether it's a molecule or whether it's some kind of a very crude preparation of a material. So adjuvants are substances which, when mixed with an immunogen or antigen, a better immune response is obtained from the host. In practical terms, higher amount of antibodies are obtained in the antiserum. Jules Freund described water in mineral oil emulsions. Herbert in 1968 confirmed that antigens injected with such an emulsion persisted up to 544 days. On the other hand, oil in water emulsions were nowhere good. Such emulsions are called Freund's incomplete adjuvant. There is a complete Freund's adjuvant also described, which additionally has dried heat-killed bacteria such as Mycobacterium tuberculosis. The presence of glycolipid in this also enhances cell-mediated immunity and activity of the other associated immune system members such as macrophages. The mechanism by which adjuvants enhance immune response varies from adjuvant to adjuvant. What is shown here is actually a very general picture. In general, it helps to obtain higher responses with respect to the both humoral and cell mediated responses. Hence, the use of the adjuvants is very common whenever we are trying to raise anti-serum in experimental animals or even during vaccination, as we will see. Adjuvants play a very important role. Let us look at the list of some commonly used adjuvants and get some idea about the mechanisms by which they operate. Essentially, they help in the increase in antigenicity of the antigen which is being injected. Freund's complete adjuvant, which is listed there, is not used in humans as it produces chronic inflammation around emulsion depots. Many adjuvants are specifically used for stimulation of pathological condition in animals. For example, complete Freund's adjuvant can induce autoimmunity. Finally, many non-immuno-enhancing drugs can also be used as adjuvants. For example, levamisole, isopronosine, and avridine. The simplest adjuvants are compounds of aluminium and calcium, beryllium sulfate. The compounds of the aluminium and calcium and beryllium sulfate, they basically stimulate the cells. Mycobacteria, when used as an adjuvant, facilitates antigen processing and T cell response. Bordetella pertussis is also used as an adjuvant, and it increases response in terms of B and T cells as well as in terms of the macrophages. Bacterial lipopolysaccharides, when used as an adjuvant, increase B cell activity. Many other substances like saponin, lysolecithin analogues, even vitamins A and E, some polyanions and liposomes, they are supposed to work by as an adjuvants by facilitating antigen processing. We refer to the formation of the precipitate between soluble antigens and antibodies. Let us look at this reaction, which is called the precipitin reaction. In this case, the antigen molecules combine with the antibody molecules in a serum within few minutes. When the univalent heptanes are used as antigens, these antigen antibody complexes remain in soluble forms. Similarly, if monovalent antibody fragments FAB are used in place of antibody molecules, we get these soluble immune complexes. However, if the antigen is multivalent and the antibody, which normally at least is bivalent, they are used, then after about 12 to 24 hours, a precipitate becomes visible. This kind of a reaction can be done even in a test tube. This precipitate results from the formation of a lattice-like structure. In the figure, you can see that how these networks arising from the cross-linking between these antibody and antigen molecules arises. At the equivalence zone, the precipitate is maximum. In this equivalence zone, most of the antigen and antibody molecules are part of this precipitin complex and do not exist free. Away from this zone of equivalence on both sides, initially when you start adding antigen, there is a zone of antibody excess. So all the antibody is yet to be tied up in the formation of these immune complexes. As you cross the zone of equivalence, which is done when you go on adding antigen, we cross over to the zone of antigen excess. This is the zone where now some of the antigens exist and are free and are not a part of the precipitin lattice. This precipitin reaction was discovered long back and is still considered a very valuable test for testing the presence of antigens or antibodies. This figure shows how actually it looks when carried out in a test tube. The precipitation of the immune complex was studied by Cross in 1897. Cross also established that this test works only with homologous antiserum. This technique can measure the potency of different antisera against a given antigen. It can also establish relative heterogeneity of the given antigen-antibody pairs. The precipitate can be weighed after centrifugation, and thus the method can be adapted to some kind of a quantitative format. The mechanism of precipitate formation was studied by Merak, Heidelberg and Kendall. The interlocking three-dimensional lattice after a critical size cannot remain in solution. Sedimentation rate of the lattice is proportional, is shown here in this equation, where V equal to volume of the complex, rho is the density of the particle or complex, and rho 0 is the density of the solvent medium. Protein antigens in the molecular weight range of 40 kilodalton to 160 kilodalton tend to result in a precipitin curve with a sharp peak. Denatured proteins, some viruses and polysaccharides, on the other hand, tend to result in a precipitin curve with a broad maximum. Amino acid composition at the antigen binding site of the antibody plays an important role in forming this lattice. Similarly, highly charged antigen molecules disrupt this lattice formation due to electrostatic repulsion and do not form well-defined precipitin curve. Audin in 1946 demonstrated that precipitin reactions can be carried out in agar gels. What really done is that the agar is plated on a petri dish. Wells containing antigen molecules and wells containing antibodies against the antigen are created. Antigen and antibody molecules can diffuse through the semi-solid agar gel and give precipitin reaction at a point where their relative concentrations fall in the equivalence zone. The lower part of the figure shows the well containing 4 different types of antigens and the well containing different types of antibodies, and hence one can see the multiple precipitin lines at different distances between the two wells. Let us look at the experimental part in a little bit more detail. So, as we said, agar gels are poured into these petri plates and allowed to set. Generally 1 to 2% agar in buffer at pH 7 to 8.5 is used. Wells are then punched into the gel and the test solutions of antigen and antibody are added. Antibody is generally placed in the center. The solution diffuses out, and where antigen and antibody meet, they bind to each other. They cross-link and precipitate, leaving a line of precipitation. Depending upon the different types of antigens which react with the antibody, different lines of precipitation emerge. The double diffusion method developed by Octoloni is very useful in establishing the identity and non-identity in antigen and antibody samples. This double diffusion technique may be used to determine the relationship between antigens and a particular test antibody. 3 basic patterns which can emerge are shown here. In the case A, which is known as the reaction of identity, the precipitin arcs form between the antibody and the two test antigens fuse, indicating that the antibody is precipitating identical epitopes in each preparation. In the B case, which represents the case of non-identity, the antibody recognizes the two different antigens and develop two different precipitation lines which intersect. The C case represents a situation of partial identity wherein the antibody recognizes that the two antigens share the same epitope, but the second antibody also has an additional epitope, thus a spur appears. Another widely used technique, a simple technique in immunology, is the technique of immunoelectrophoresis. Immunoelectrophoresis was developed by Graeber and Williams in the 1950s. It's very useful clinically. The antibody concentration in the range of the 3 to 20 microgram per ml of the antiserum can be detected qualitatively. Immunoelectrophoresis as a technique essentially has two steps. First, the various antigens in the sample are separated by carrying out the electrophoresis in agar gel. In the second step, double diffusion analysis with the corresponding sera containing antibody for various antigens is carried out. The result normally appears typically in the way it is shown here. Antigens move with different velocities in the electric field depending upon their E by M ratio. These separate bands of antigens then diffuse towards the diffusing antibodies and form the precipitin bands, which can be clearly seen in this figure. There is a variation of immunoelectrophoresis which is called rocket electrophoresis. This is also called Laurel technique. It is a single step method. As the antibody containing the antiserum is incorporated in the agar gel itself, the pH is chosen so that the antibodies contained in the gel are immobile and the antigens are negatively charged. The precipitin arcs have the shape of rockets, and that is why this technique is called rocket electrophoresis. The height of the rocket is proportional to the antigen concentration. The sensitivity of this method is about 10 to 20 times higher than the simple double diffusion method. There is another interesting variation of the rocket electrophoresis which involves slight modification. This is called two-dimensional electrophoresis. In this, antigens are separated by electrophoresis. After blotting, these separated antigen bands are used to carry out rocket electrophoresis in another dimension. So this involves the use of the blotting technique as one of the intermediate step. We had referred to agglutination earlier as a phenomenon briefly. Let us look at that again in slightly more detail. If the antigens are in the form of cells such as RBCs, or antigens can be converted into a particles by immobilizing them on the latex beads. This kind of a immobilization can be carried out in numerous ways. The antigens can be bound to latex beads by simple adsorption. In case it becomes necessary, the typical conjugation reactions which are known in the protein chemistry. They can be used to covalently link the antigens to these latex particles. So, whether we are using RBCs or antigens bound to these particles, now the antigen are in the particulate form. The reaction between these multivalent antigens and bivalent or multivalent antibody forms clumps. Should be obvious that the clump formation is maximum in the equivalence zone. There is a quantitative version which generally employs serial dilution. In this serial dilution technique, we determine the maximum dilution of the antiserum at which the clump formation can be detected. While it is kind of semi-quantitative, this is used widely to gain roughly some idea about the amount of the or the concentration of the antigen or antibody in these samples. The clump formation in these agglutination tests is the result of the agglutination of the antigen via cross-linking with the antibody molecules. So, the highest dilution at which agglutination or the clump formation is observed is called the titer value. As we mentioned, these agglutination tests are not completely quantitative, but these are sensitive enough to detect antibody concentrations of as low as 0.001 microgram per ml of the serum. These agglutination reactions are popular because they are simple, rapid, and they provide a very sensitive method of identifying types of RBCs, bacteria, fungi, and other cells. So students, let's now summarize what we have learnt in this module. To summarize this lecture, we learnt about role of adjuvants in immunization. We learnt about the precipitin reaction in solutions. We learnt about the precipitation reactions on agar gels. We learnt about the immunoelectrophoretic techniques, and we learnt about the agglutination reactions. In this lecture, all the techniques were based upon so that all can be carried out using antibody in the form of antiserum. All techniques essentially relied upon precipitation of an antigen with an antibody complex in free solution or on agar gels. Thank you.